Welcome. This is the third lecture on enzyme. Third lecture on the enzyme. We are discussing one after another. Now, today we'll discuss about the first. We'll discuss about the importance of game. Game, the Michaelis constant, is equal to the substrate concentration at which the reaction rate is half its maximal value. The Km value of an enzyme depends on the particularly substrate and environmental conditions such as pH, temperature, and ionic strength. The Michaelis constant Km has two significances. First, Km is the concentration of the substrate expressed in moles per liter at which half the active sites of the enzymes are filled. Thus, Km provides a measure of the substrate concentration required for significant catalysis to occur. Km is independent of enzyme concentration. Second, Km is a measure of the strength of the ES complex, enzyme substrate complex, or we can say it is a measure of the affinity of the enzyme for its substrate. Affinity of the enzyme for its substrate. It reflects a small Km, number A or number 1. Small Km reflects high affinity of enzyme for substrate. And the large Km or high Km reflects low affinity of enzyme for substrate. Indicates weak binding and low affinity of enzyme for substrate. Some examples are carbonic anhydrase. This is the name of the enzyme. Substrate carbon dioxide. Km value is 8000. Whereas lysozyme enzyme, substrate N-acetyl glucosamine, the Km value is just 6. Similarly, beta-galactosidase enzyme, substrate is lactose, Km value is 4000. Next, Lineweaver bark plot or double reciprocal plot. You see the picture. Lineweaver bark plots are used to obtain values for Vmax and Km. A more accurate method of determining values for Vmax and Km uses Lineweaver bark equation shown. The equation is obtained by taking the reciprocal of the Michaelis-Menten equation. 1 by V0 equal to Km by Vmax into S plus 1 by Vmax. When 1 by V0 is plotted against 1 by S, a straight line is obtained with a slope equal to Km by Vmax. The point at which the line intersects the y-axis is numerically equal to 1 by Vmax, and the point at which the line intersects the x-axis is numerically equal to minus 1 by Km. Significance of Lineweaver bark plot: the values of Km and Vmax can be readily calculated from the values of these two intercepts, and also the plot is useful to determine the mechanism of action of enzyme inhibitor. Importance of Vmax. Number 1. Vmax or maximal rate reveals the turnover number of an enzyme, which is the number of substrate molecules converted into products by an enzyme molecule in a unit time when the enzyme is fully saturated with substrate. I repeat, turnover number is the number of substrate molecules converted into products by an enzyme molecule in a unit time when the enzyme is fully saturated with substrate. Second one, Vmax of a reaction is an index of the catalytic efficiency of an enzyme. The Vmax is useful in comparing the activity of one enzyme with that of the another. And third one, comparing activity of an enzyme with another. As for example, carbonic anhydrase, it has got turnover number 60,000 per second, whereas lysozyme just 0.5 per second. Turnover number is equal to rate of constants K2. Turnover number is equal to rate of constants K2 of the reaction I have shown you already. Mechanism of enzyme action. Two models for substrate binding to the active site of the enzyme have been proposed to explain the specificity that an enzyme has for its substrate. A. Lock and key model or rigid template model of Emil Fischer, and B. Induced fit model or hand-in-glove model of Daniel E. Kosland. We will discuss one by one. Lock and key model or rigid template model of Emil Fischer. You see the upper part of the picture. In this model, enzyme is pre-shaped and the active site has a rigid structure that is complementary to that of the substrate. This model is called lock and key model because in this model the substrate fits into the active site in much the same way that a key fits into a lock. This model has been useful in understanding how some enzymes can bind only a specific substrate but will not bind another compound with an almost identical structure. For example, most enzymes in carbohydrate metabolism can bind the D isomer of hexoses but cannot bind the corresponding L isomer, which differs only in the configuration around a single carbon atom. This model explains all mechanisms but do not explain the changes in the enzyme activity in the presence of allosteric modulations. B. Induced fit model. In induced fit model or hand-in-glove model of Daniel E. Kosland. B. Induced fit model of Daniel E. Kosland. In Fischer's model, explained the specificity of enzyme-substrate interaction, but the implied rigidity of the enzyme's active site failed to explain the dynamic changes that must take place during catalysis. In the Fischer's model, the catalytic site is presumed to be pre-shaped to fit the substrate. However, Daniel E. Kosland postulated that the enzymes are flexible and shapes of the active site can be modified by the binding of the substrate. So, in the induced fit model, the substrate induces a conformational change in the enzyme in the same manner in which placing a hand substrate into a glove enzyme induces changes in the glove shape. Therefore, this model is also known as hand-in-glove model. Conformational change in enzyme aligns catalytic residues which participate in catalysis. The enzyme in turn induces reciprocal changes in its bound substrate that alters their orientation and configuration and strains the structure of the bound substrate. Such change helps to bring the enzyme substrate complex into its transition state. The intrinsic binding energy due to substrate enzyme interaction is made available for the transformation of the substrate into product. This model is believed to describe more accurately the specificity of substrate binding than do lock and key model of Emil Fischer. And this is, however, is the accepted model. Our next topic is enzyme regulation. Now, first of all, in each enzyme system, there is at least one enzyme that regulates the rate of overall metabolic sequence as per the cell's demand. Such regulatory enzymes not only have catalytic function but are also capable of increasing or decreasing their catalytic activity in response to certain signals. Such enzymes, whose activity is modulated through various types of molecular signals, are called regulatory enzymes. In most multi-enzyme systems, the first enzyme of the sequence is the regulatory enzyme. There are four major classes of regulatory enzymes, or there are four major processes of enzyme regulation. First one is allosteric enzymes, allosteric or non-covalently regulated enzymes. Second, covalently regulated enzymes. Third, enzymes regulation by induction and repression of enzyme synthesis. And fourth, by regulation of enzymes by proteolytic cleavage. So we will discuss one by one. First one is the allosteric enzymes. The term allosteric derives from Greek word allo equal to other and steros or space or site. Allosteric enzymes are those having other site. Like the enzymes, allosteric enzymes have catalytic sites which bind the substrate and transform it, but they also have one or more regulatory or allosteric site for binding regulatory molecules, which is called effector or modulator or modifier. You see the picture in the downwards part of the shown OHP sheet. The allosteric enzymes may be inhibited or stimulated by their effectors or modulator. Effectors that inhibit enzyme activity are termed negative effectors, whereas those that increase enzyme activity are called positive effectors. I believe from the picture you will be able to understand the mechanism of allosteric enzyme. Here NM stands for negative modulator, PM for positive modulator, S for substrate, A for allosteric site, C for catalytic site, and ES equivalent to enzyme plus product. In this picture, you see the effect of substrate concentration on reaction velocity for an allosteric enzyme. Just as the catalytic site of an enzyme is specific for its substrate, the allosteric site is specific for its modulators. Allosteric enzyme molecules are generally larger and more complex than those of simple enzymes. Most of them have two or more polypeptide chains or subunits. Allosteric enzymes do not obey the Michaelis-Menten behavior. A sigmoid saturation curve results rather than the classical hyperbolic substrate saturation curve shown by non-regulatory enzyme. Second one, covalent regulation of enzyme. Some enzymes are regulated by reversible covalent modification, most frequently by addition of phosphate groups, that is phosphorylation, or removal of phosphate groups, dephosphorylation, from specific serine, threonine, or tyrosine residues of the enzyme, as shown in the lower part of the picture. Depending upon the specific enzyme, the phosphorylated form may be more or less active than the dephosphorylated enzyme. For example, phosphorylation of glycogen phosphorylase An enzyme that degrades glycogen to glucose increases activity, whereas the addition of phosphate by glycogen synthase, an enzyme that synthesizes glycogen from the glucose, decreases activity. I have shown this example in the lower part of the picture. The third, induction and repression of enzyme synthesis. The increased, that is induction or decreased repression synthesis of the proteins, enzymes, leads to an alteration in the total population of active sites rather than influencing the efficiency of existing enzyme molecules. Enzymes subject to regulation of synthesis are often those that are needed at only one stage of development or under selected physiological conditions. For example, elevated levels of insulin due to high blood glucose levels cause an increase in the synthesis of key enzymes involved in glucose metabolism. In contrast, enzymes that are in constant use are usually not regulated by altering the rate of enzyme synthesis, and alteration in enzyme levels due to induction or repression of protein synthesis are slow, hours to days, compared to allosteric changes in activity which occur in seconds to minutes. The fourth type of enzyme regulation, that is regulation of enzyme activity by specific proteolytic cleavage. Some enzymes are synthesized in inactive precursor form, that is called zymogen or proenzyme, that are subsequently activated by cleavage of one or few specific peptide bond. An energy source ATP is not required for cleavage. In contrast, with reversible regulation by phosphorylation, even protein located outside cells can be activated by proteolytic cleavage. Another difference is that proteolytic activation, in contrast with allosteric control and reversible covalent modification, occurs just once in the life of an enzyme molecule. Specific proteolysis is a common means of activating enzymes and other protein in biological system. For example, activation of the digestive enzyme that hydrolyze proteins which are synthesized as zymogens in the stomach and pancreas. Second example is enzymes of blood clotting. Similarly, third example is formation of the fibrous protein collagen from procollagen. In zymogens of digestive enzymes that hydrolyze protein, some examples are zymogen pepsinogen, active from pepsin, site of synthesis stomach. Chymotrypsinogen zymogen, active from chymotrypsin, site of synthesis pancreas. Trypsinogen, that is the zymogen, active form is trypsin, site of synthesis pancreas, and like that.